Welcome to Biochemistry. Today, we are going to explore exactly how our bodies handle the proteins we eat. We will uncover the fascinating and essential processes of transamination, deamination, and the urea cycle. By the end of this lesson, you will understand how we survive on a high-protein diet. This is a fundamental concept for anyone studying zoology or metabolic processes, as it explains the delicate balance of life at a cellular level. To start, imagine you are trying to recycle an old, broken-down car to use its metal for building something new. Before you can safely melt down the steel chassis, you absolutely must remove the toxic lead-acid battery, or else the entire recycling factory will be contaminated. In the human body, dietary proteins and cellular amino acids are exactly like that car, and their nitrogen-containing amino groups are the toxic batteries. We cannot simply burn amino acids for energy because releasing free nitrogen directly into the bloodstream forms ammonia, which is highly toxic to our cells and can cause severe neurological damage. Because free ammonia is so dangerous, the body has developed a specialized two-step biological recycling system. First is transamination, a crucial step where the amino group is transferred from an amino acid to a keto acid. This does not remove the nitrogen, but rather funnels it into a single carrier molecule called glutamate. Next comes oxidative deamination, which takes place primarily within the mitochondria of the liver. Here, the amino group is finally stripped away from the glutamate carrier and released as free ammonia in a highly controlled environment, ensuring that nitrogen is safely gathered without poisoning the surrounding delicate tissues. Now that we understand how nitrogen is processed, let us look at how we measure it mathematically in the body. This is the nitrogen balance equation, which conceptually tracks what goes in versus what comes out. The symbol Bn represents the overall nitrogen balance in the body. The variable In stands for the total nitrogen intake derived from dietary protein. The variables Un, Fn, and Sn represent the nitrogen lost through urine, feces, and sweat, respectively. If the intake increases while excretion remains constant, the balance becomes positive. Physically, a positive balance indicates that the body is actively building new tissue or muscle. Let us see this with a concrete example. A recovering hospital patient consumes exactly 120 grams of protein in a single day to help rebuild damaged tissues. We need to find their nitrogen balance to see if they are healing. First, we must calculate the total nitrogen intake. Since average dietary amino acids contain about 16% nitrogen by weight, we multiply the 120 grams of protein by the constant 0.16. This gives us an intake of 19.2 grams of pure nitrogen. This is the total amount of raw nitrogen material the body has available for metabolic processes that day. Building on our intake calculation, we now apply the primary nitrogen balance equation to find the final result. The laboratory tests show that the patient excretes 14 grams of nitrogen in urine, 2 grams in feces, and loses 1 gram through sweat. We subtract this total combined loss of 17 grams from our 19.2 grams of ingested nitrogen. The mathematical result is a positive 2.2 grams. Physically, this positive value means the patient is retaining more nitrogen than they are losing. It indicates that the transamination pathways are actively using the retained amino acids to synthesize new structural proteins. While retaining nitrogen is great for healing, breaking down proteins for energy creates a massive problem. what happens to the toxic ammonia generated during deamination? When the body breaks down amino acids, it produces free ammonia. This ammonia is as dangerous to our internal cells as raw sewage is to a city's water supply. The body cannot simply hold on to this ammonia, nor can it easily transport it through the blood. If it builds up, it crosses the blood-brain barrier and causes severe, irreversible damage to the delicate tissues of the brain. To solve this dangerous problem of ammonia buildup, the liver acts as a highly advanced biological wastewater treatment plant. It employs a brilliant metabolic pathway called the urea cycle. You can think of the cycle as a chemical incinerator. Through a continuous series of 5 biochemical reactions, it systematically takes 2 molecules of highly toxic ammonia and safely packages them with carbon dioxide into a single molecule of urea. This new compound is harmless and water-soluble, allowing it to be easily flushed away by the kidneys without damaging the brain. While the urea cycle is absolutely essential for survival, it is not free. It is a highly endergonic process, meaning it requires a significant input of cellular energy. This formula calculates that energetic cost. The variable E sub C represents the total energy cost, measured in the number of high-energy phosphate bonds broken. The variable N sub U represents the number of moles of urea produced. We multiply by the constant 4 because each cycle consumes the equivalent of 4 high-energy phosphate bonds. This formula clearly shows that as the production of urea increases, the energy burden on the liver increases linearly and significantly. Let us apply this cost formula to a real-world scenario to see the impact. A bodybuilder consumes a massive amount of protein, forcing the liver to synthesize 2.5 moles of urea in a single day to clear the excess nitrogen. To find the total energetic cost, we multiply the 2.5 moles of urea by the 4 high-energy phosphate bonds required per mole. This means the liver must expend the equivalent of 10 moles of ATP just to safely package the toxic ammonia generated from their diet. It highlights how metabolically expensive it is to use protein as a primary fuel source. Now that we have the total ATP cost, let us see what this means for the body's carbohydrate reserves. We take the total energetic cost of 10 moles of ATP equivalents and divide it by 32, which is the standard net yield of ATP from the complete cellular respiration of 1 molecule of glucose. The result is 0.3125 moles of glucose. Physically, this shows that a significant portion of the body's energy is being diverted away from muscle building or movement. Instead, this precious glucose is being spent purely on waste management to clear out the toxic nitrogen. To fully grasp how this expensive process works, let us look at the urea cycle pathway visually. Notice how the cycle spans across 2 distinct cellular compartments. It begins inside the mitochondrion, where ammonia, carbon dioxide, and ATP combine to form carbamoyl phosphate. This feeds into the cycle, producing citrulline, which is then transported out into the cytosol. Through subsequent steps involving aspartate and ATP, arginine is formed. Finally, arginine is hydrolyzed to release urea and regenerate ornithine, keeping the cycle turning continuously. This diagram perfectly illustrates the coordinated effort required to manage dangerous waste. We have covered a lot of ground today, so let us summarize the most important ideas. First, transamination and deamination work together to safely remove toxic nitrogen from amino acids without poisoning the body. Second, Tracking nitrogen balance tells us if the body is actively building or breaking down tissue. Finally, the urea cycle is a vital but highly energy-intensive process that converts toxic ammonia into harmless urea. You now have a solid understanding of how our bodies manage protein metabolism and the profound consequences of our dietary choices.